Okay, so in this one we're going to have a kind of in-depth look on antibodies, the things produced by the B cells of the adaptive immune system that bind to pathogens, and we're going to talk about why they do that and what they're doing, and the structural components of antibodies. So, getting into it. Antibodies are basically these things called immunoglobulin proteins. You can use the terms interchangeably, which we're going to do. So don't get confused. An immunoglobulin is just the kind of proper word for an antibody. So produced by B cells, antibodies are highly specific proteins called immunoglobulins, capable of recognizing a vast array of antigens. They can exist either in an attached form to the membrane of B cells or mast cells, or in a secreted form where they kind of exist independently of any kind of cell and just float around the blood or the lymph or in the tissues. So an antibody has a variable region that is complementary to a small section of a pathogen called an epitope. So if you look to the image on the right, you can see the parts at the tip of the kind of Y shape of the immunoglobulin molecule that are coloured in kind of purpley pink. They're called the variable regions. The tips of these variable regions are called the antigen binding site. So basically, these are the things that are going to bind to the antigen. And this thing, the epitope, that just refers to the part of the antigen that the antigen binding site of the variable region is actually complementary to in shape, and so it's going to bind to it. Each immunoglobulin is made of 4 polypeptide chains, and these are 2 identical light chains and 2 identical heavy chains. So in the image on the right you can see the heavy chains shown in blue, and the light chains shown in red. And then the extension bits are the variable regions. They're still part of the heavy and light chains, but they're just subject to a lot of diversity so that they can have different kind of shapes, so that the immunoglobulins can have antigen binding sites that can recognize pretty much any kind of epitope they come across, so they can kind of recognize any potential antigen that they might need to bind to. So they can basically have binding sites. It's so diverse they can kind of recognize anything, but obviously each one is different, so only one kind of immunoglobulin is going to be able to bind to one kind of antibody. So the heavy chains and light chains both have a variable region, like we talked about here, shown in pink, and a constant region, which would be this part in red and this part in blue for the light and heavy chains respectively. So the amino acid sequence in the variable region of these immunoglobulins is different in every single immunoglobulin molecule, and it's this variation in the amino acid sequence that allows B cells to essentially produce immunoglobulins with specificity to any kind of potential pathogenic epitope. So it's kind of put in fancy words, but it just means that the variable regions are going to come in like so many different shapes that a specific one is going to be able to recognize like a specific antibody no matter what. So imagine an antibody is round, you might get like a lot of square antigen binding sites, but because it's so diverse, you're also going to have round antigen binding sites on these immunoglobulins that would pick up that specific antigen, and then you'd have your antibody-antigen complex. The constant region plays no part in this antigen recognition and epitope recognition, but it is responsible for determining the isotype of the immunoglobulin. And the isotype reflects the immunoglobulin's biological function. So shown on the bottom right here, you can see the different kinds of immunoglobulin isotypes: IgG, IgE, IgD, IgM, and IgA. They all have their own functions that we're going to talk about in a minute. We'll just get onto it. So immunoglobulin isotype is determined by the constant region of the heavy chain. So this blue region here is determining the isotype of the specific immunoglobulins. Immunoglobulins can be made of 5 different kinds of heavy chain, determined by these different genes shown on the bottom here. So you can see there are 5 different colors, excluding grey, which is, I think, coding for receptors on the heavy chains that are involved with the natural killer cell recognition of immunoglobulins at the Fc region that we're going to talk about in a minute. But you can see that they come in 5 different colors, controlled by these different genes here. And this each one of these kind of colored blocks is going to produce a different type of isotype. So you can see there are 5 different isotypes, like we talked about a minute ago, all shown in this image here. And you might wonder why there are 4 yellow blocks and 2 orange blocks. Well, they all belong to the same isotype, but we can further divide isotypes into subtypes. So you can see I've written here that there are 4 subtypes of immunoglobulin G. So yeah, you can see 4 subtypes of immunoglobulin G. Each subtype is going to be determined by which one of these 4 kind of genes is responsible for coding for the heavy chain. And then the same thing going for immunoglobulin A. You can see it's represented in orange here. Either subtype is going to be determined by which of these 2 orange genes are going to be coding for the constant region of the heavy chain. So with regards to the functionality of these different isotypes, it can actually change their structure. So immunoglobulin A can form a dimeric structure with a J chain, so it means 2 IgA molecules can bond together with help of a J chain, shown here with the little grey circle. And IgM can actually form a pentameric structure with a J chain in the middle with 5 immunoglobulin M molecules. Then all the other isotypes are singular. The structure of these isotypes is going to alter their function and what they can actually do. So antibodies bind to the epitope that their variable region is complementary to. This binding marks the pathogen for phagocytosis by phagocytes, a process we call opsonization. Antibodies' Fc portions on the bottom half of the chain shown in this picture on the right can have receptors that permit natural killer cells to bind to them to trigger antibody-dependent cell-mediated cytotoxicity, which we went over when we were talking about T cells. Antibodies can also facilitate a process called complement activation. I've just put an example there about an example of an opsonizing complement called C1q. And also antibodies can be secreted to bind pathogens, to neutralize them, agglutinate them, precipitate them, but remember, function depends